Good evening. We will start in a couple of minutes. I hope the audio is clear and also the screen is presented and visible. Okay, let's start the session. Good evening. Welcome you to the live practice session, week 9 for the NPTEL course on environmental biotechnology. In continuation to the previous session, we'll be seeing about bioremediation, focusing on biodegradation. So when we talk about bioremediation process, um, the, the science and the technology has developed over a long period of time now. So the evolution of bioremediation approaches in terms of environmental biotechnology we'll be seeing now. The first phase is the pre-genomic phase. So, in the pre-genomic phase, it encompasses both non-molecular and molecular approaches that were developed before whole genome sequencing. So, whole genome sequencing is taken as the landmark, and we have pre-genomic and post-genomic phases. In the pre-genomic phase, deals with both non-molecular and molecular approaches, and in this phase, the isolation and characterization of microbes was focused. So, and also, by talking about microbes, we have bacteria, archaea, algae, fungi. But in the pre-genomic phase, isolation and characterization, especially of bacteria, was considered more. And we all know that people go for this molecular approaches to study to study the ecology ecosystem in a particular habitat, all for different purposes. One is to understand how the natural habitats function, why do a place have a certain characteristics, what is the resources available, and what microbial action causes production of some products. So all this was one phase of understanding. So the application part that is derived from that understanding is what we are now discussing as bioremediation. So in this, people studied about the environment to develop some processes that can act as bioremediation approaches. So this is because they saw some pollutants and found the importance to treat the particular site. So in that point of view, the one thing that bridges this scientific understanding and the technological application is the uh, biotechnological study. So in that context, now we are seeing about the pregenomic phase. So we now know that the pregenomic phase focused on both non-molecular and molecular approaches. So when we're talking about the non-molecular approaches, the non-molecular approaches were dominant initially due to the lack of adequate molecular biology tools. And in while Okay. And the non-molecular approaches involve direct isolation of bacteria from contaminated environments. Like people go for some culturing technique. So they isolate a particular soil, try to extract the bacterium or the microorganisms, try to grow them in laboratory conditions, which we call as culturing, and then study the organism. So the first important thing in this non-molecular approaches is the direct isolation of bacteria. And then after that, people studied the metabolic and ecophysiological characterization of the potential candidate organisms. They identified major metabolic activities and catalytic activities. And then went on scrutinizing the essential nutrients and environmental conditions for the organisms to survive. So the process was very simple, as told. I'll just reiterate. First, people go to a particular site. So the scientists collect the sample, they do the direct isolation of bacteria, and then go about studying the metabolic and ecophysiological characteristics of that microorganism. And by that, they now identify the metabolic and catalytic activities. So after this, they also try to scrutinize what is the optimum nutritional and environmental condition required for the growth of that organism. And based on this knowledge, they try to use that organism or a group of organisms for bioremediation processes. But as we all know that this process has its own disadvantage, that um, the performance that the organism gives in the lab may not be the same in the real contaminated site. So that is what is mentioned as the in situ performance. So laboratory performance and in situ remediation site performance are two different things. We may not get the replicability because in laboratory conditions we have a very controlled environment where we supply all the required nutrients. But in the natural habitat, there may be some environmental condition shift or deficit in nutritional requirements. So in that case, whatever we have hypothesized will not be working in real-time conditions. So that is one of the major drawbacks of this non-molecular approaches. Coming to the molecular approaches, it started with the advent of polymerase chain reaction, and scientists developed a technique called cloning, and then there was great advancement in sequencing, starting from Sanger sequencing to next generation sequencing. The advancement in sequencing was also very wide. So molecular approach ideally included PCR cloning and sequencing technologies. So we know that these molecular approaches were primarily designed based on the 16S ribosomal RNA because it has both conserved and variable region. Also, polyphasic taxonomical studies were done, like FAME analysis, fatty acid methyl ester analysis, lipid markers were studied, quinone signaling was studied, and analysis of single copy marker genes were done. So based on this, scientists tried providing a taxonomic affiliation for the microorganisms. And then genetic analysis of the isolated strains were performed. This is because of the advent of cloning and sequencing. And monitoring of the target organism in real environments were done. Like we all know, we collect the environment sample without the need for culturing. We go upon analyzing the genetic nature of the microorganism. And apart from that, apart from the genetic studies, people also used chemical analysis to study the fate of contaminants, because when we have to study degradation, we should make sure that the compound has degraded. So for that, people use techniques like chromatographic techniques like gas chromatography, mass spectrometry, which is even widely used in the laboratories now. Apart from that, we have techniques like high performance liquid chromatography and mass spectrometry. So these are other chromatographic techniques that we can use to trace the fate of contaminants. So we can find out how the microorganism is degrading a particular contaminant and what is the product that is produced. So we should make sure that the degradation product is not more harmful than the original contaminant itself. So, and also we have to elucidate the mechanism of degradation also. So for all these purpose, we go for the chemical analysis. So this is about the non-molecular and molecular approaches in pre-genomic phase. In the post-genomic phase, after the advent of whole genome sequencing, especially the high throughput NGS sequencing, um, the era of post-genomic phase started. The primary advantage of the post-genomic phase is it gives a complete understanding of the microbial function and its response to a particular environment. It responds to the physical conditions like temperature, pressure, and also to the chemical is present in the environment like the contaminants itself. And also we can know the activity during the bioremediation. Another advantage is that it also tells us about the hidden genetic capabilities. So genetic capabilities means the genes are not expressed. So if they are not expressed, we cannot find it out visually. For example, as it is given here, flagella genes in apparently non-motile bacteria. So a bacteria can be non-motile, which is not movable. But when we do a genetic analysis, we still find the genes of flagella present in the genetic makeup of that organism. So we can come to know the hidden genetic capabilities of microorganisms, and further using molecular studies we can try to explore or express the hidden genetic capabilities also based on um, our requirements. And again, the post-genomic phase provides a better characterization of organisms individually and as community members. So this is the key point. So in the pre-genomic phase, we were mainly dependent on culturing or a single bacteria isolation. So that is why we had a major drawback that the activity of the microorganism is not very highly likely to be replicable in in situ environments. So we need to know how the organism behaves individually to perfectly characterize it, but we equally we need to know about its interaction with other microorganisms in the uh, habitat. So this is the ecological analysis. So we are studying the interactions between the organisms. So that is possible in the post-genomic phase. And next comes the advantage that it allows development of conceptual models for bacterial functioning. So post-genomic phase, as we know, gives us high throughput uh, data. So we get a large amount of data. We are also developing computational tools to process those data. So as a result, we have more data which we use to build models, and then these models help us in predicting the ecosystem behavior. So these are the main advantages of the post-genomic phase. But there are also few limitations of the post-genomic phase, which requires cultivable organisms. We know the great plate count anomaly. So like around only 1% of the bacteria are culturable. So that is the great plate count anomaly. So that is one of the main limitations in the post-genome phase. and the risk of isolating organisms that are not dominant or influential in their natural environment, which is more or less similar to the non-molecular approach. And then we need to pay careful attention to the culture conditions and environmental parameters, otherwise we may lose some of the key organisms that are present in the consortium. And the revolutionary progression demonstrates the increasing sophistication in understanding microbial communities and their applications in environmental remediation. So the terminal, so the next we will be seeing some of the terminal electron acceptors in bioremediation. So terminal electron acceptors play a crucial role in the oxidation of toxic pollutants, which remains one of the most common types of biodegradation. So in biodegradation, we have many kinds of reactions happening. The most prominent one is the redox reaction. So when we talk about redox reaction, there is going to be electrons involved in it. So loss of electrons, gain of electrons, all of this is going to happen, and that is why we have um, the redox reactions coming into picture. And this electron loss or gain transforms the pollutants, or and in many cases mineralizes the pollutants. So in this case, terminal electron acceptors are key components in bioremediation process. So electron, so coming to the electron transport process, when organic pollutants undergo microbial catalysis, microorganisms can oxidize the organic compounds, which causes the complete degradation. And here, carbon dioxide and water are produced along with energy release. So, this complete degradation is known as mineralization. And this energy is utilized by the microbes for growth and proliferation. So, they use these pollutants as substrate for producing energy, and that energy is used for growth and proliferation. So the mechanism of this electron transport goes like, first the oxidation of organic carbon releases the electrons, and these electrons are transported through electron carriers. And we also have generation of reducing powers like NADH and FADH2. And from there, the donation of electrons to membrane bound electron carriers is there, and final transfer to the terminal electron acceptor happens. So, this is how the electron is transferred from the substrate to the terminal electron acceptors through electron carriers and reducing powers. So we have different types of terminal electron acceptors. So first one is the in the aerobic condition. So in aerobic condition, the main component is oxygen. So oxygen serves as the primary terminal electron acceptor in aerobic bacteria, and this is well studied in oxic habitats. So when we talk about aerobic, it is oxic; anaerobic is anoxic. Aerobic directly involves air, in which oxygen plays a crucial role. From the other components of air, oxygen serves as the primary terminal electron acceptor. So in order to have oxic habitats, we need the polluted surface to be exposed to air, either naturally or while designing a process. We have to design such that we uh, supply oxygen or air for aerobic degradation to take place. So in nature, when we see petroleum, oil and hydrocarbon contaminated sites are naturally prone to oxygen, and here aerobic degradation can happen, where oxygen will serve as the terminal electron acceptors. So some examples of organisms that take up this aerobic route include Pseudomonas and Burkholderia. When talking about the anaerobic conditions, we have anaerobic conditions in groundwater, uh, because it is not exposed to air, other underground systems, and subsea habitats. So we all know when we have some water body, only the top surface is going to be in contact with air, and there is going to be exchange of oxygen. So only the top part is going to be aerobic. When we further move down across the depth of the water body, it is going to slowly turn into anoxic site, and here we will have the anaerobic degradation. So when oxygen is not present, nitrate, iron, sulfate, manganese, selenium kind of compounds can act as the terminal electron acceptors. And there are different kinds of microorganisms that can use each of these terminal electron acceptors. So ideally, when oxygen is present, more energy is released. So we have seen about this redox tower concept. So in that, the redox potential is very high when oxygen serves as the terminal electron acceptor, and it gradually decreases with other electron acceptors. So that is why the energy yield in aerobic processes is higher than in the anaerobic processes. So in case of nitrate, we have nitrate reducing bacteria which can convert nitrate to nitrogen. Similarly, iron reducing bacteria can convert Fe3 plus to Fe2 plus. So here you can see the redox reaction happening. And sulfate reducing bacteria um, reduces sulfur to sulfide. Apart from these three are primary, this is very widely studied and present in nature. Apart from these, components like manganese and selenium can also act as the terminal electron acceptors in anaerobic cases. So the type of terminal electron acceptors generally can be classified into aerobic and anaerobic conditions. So based on this presence of terminal electron acceptors, in contaminated subsurface environments, we can see the development of different zones. So that we call it as environmental stratification. Like there is a hierarchy coming into picture. So the surface zone, which is in interaction with air, has aerobic bacteria because it can utilize oxygen in that zone. And going to the deeper zones, we will be having nitrate, iron, and sulfate, and we can have the reducing bacteria. In that, we can find the reducing bacteria in those deeper zones. Further, if we move where even these components are not present, the deepest zones we can find the methanogenic archaea. So, methanogenic archaea can essentially use carbon dioxide to produce methane. So, this is the different zones that are developed based on the terminal electron acceptor availability. Surface zone, deeper zone, and deepest zone. In surface zone we have oxygen, in deeper zone we have nitrate, iron, and sulfate. In deepest zones we don't have any of these, but the organisms that live there can use carbon dioxide to produce methane and then still survive in that habitat. So iron is one of the preferred electron acceptors, especially Fe3 plus, because of its natural occurrence in geological strata as a part of soil and rock system. It is very abundant, and it has a higher potential to act as a electron acceptor. It enhances the Fe3 availability. If we enhance the availability of Fe3 plus ion, anaerobic degradation of organic pollutants can also be enhanced. So when we ensure the availability of proper terminal electron acceptors, proper redox conditions, and proper bacterium, we can guarantee a good biodegradation process that can happen in a particular environment. So three things that we need are terminal electron acceptors, redox conditions, and suitable catalytic microorganisms. So as we discussed, the sequence of utilization based on the redox tower or based on the presence of the components include: first, oxygen. It's the most preferred terminal electron acceptor due to its high redox potential and energy yield. Next comes nitrate. So that zone we call it as anoxic zone, particularly in subsurface aquifers and the sediments. Here oxygen is depleted. Organisms do not have oxygen here to consume, so they start consuming nitrates. And then comes manganese or iron oxides. When nitrate is unavailable, they start utilizing manganese and iron oxide. So this is based on the energy yield that the organism is getting. Oxygen provides the greatest energy yield, followed by nitrate, and then manganese and oxides. And further comes sulfate, which is present in higher, increasingly reducing conditions than the previous ones, where sulfate is transformed to sulfur. And then we have the most anaerobic condition or the deepest zone, as we saw in the previous slide, where carbon dioxide is present, and the environment is highly reducing environment where no other electron acceptors are available. Therefore carbon dioxide itself is used as the final electron acceptors. So this is the concept of terminal electron acceptors. So since most biodegradation processes deal with redox reactions, we should make sure that we understand the mechanism of the redox reactions and ensure the presence of uh, the suitable conditions for the redox reactions to happen. That is when we can see bioremediation or biodegradation taking place in a particular environment. So coming to the next part, we will also see about Geobacter species. So Geobacter species is one of the widely studied bioremediating microorganism, and it was first noticed during the bioremediation trials for uranium removal from underground contaminated aquifers. And this geobacter is capable of reducing uranium 6 to uranium 4, and because of this reduction, it can precipitate under carbonate aquifers, basically due to a pH shift due to the carbonate present at near neutral pH, making the groundwater free of soluble uranium. So the mechanism includes that the geobacter uranium reducers can utilize uranium as the terminal electron acceptors. Therefore, it reduces uranium 6 to uranium 4 plus oxidation state. But it requires organic electron donors, particularly acetate injection. So it can consume acetate, derive electrons, and then reduce uranium 6 plus to uranium 4 plus. So the extracellular reduction possible through conductive pili as a protective cellular mechanism. Apart from uranium reduction, geobacter can also go for hydrocarbon degradation, where it is capable of degrading organic carbon in anaerobic environments using iron as the iron 3 plus as the terminal electron acceptors. So this can process the aromatic hydrocarbons through iron oxide injection, where this Fe3 plus get complexed with the organic ligands and increases its solubility and bioavailability. Another interesting factor is that when both this hydrocarbon and uranium are present, geobacter species can simultaneously degrade both of them. Simultaneously remediate both of them. Degradation of hydrocarbon takes place, whereas uranium is a metal, so it cannot be degraded, but it can be removed from the system where using reduction and precipitation process. So this we call as biotransformation and not biodegradation. So now we know that Geobacter species is widely studied for its uranium remediation and hydrocarbon remediation. So in some interesting mechanisms that we know about this Geobacter is, first thing is about its motility and flagella. Initially, Geobacter was characterized as a non-motile without flagella production. So this is what we saw in one of the slides while we talked about this pre-genomic and post-genomic evolution. So when we were initially analyzing the bacterium itself, we did not find any flagella and it was termed to be non-motile. But whenever the genetic makeup of Geobacter was studied, the gene that could produce or synthesize flagella was present. So further experiments were conducted and it was found that flagella production occurs only with insoluble electron acceptors like iron oxides and manganese oxides. When there is presence of soluble electron acceptors like nitrate, there is no flagella production. So this is one of the interesting findings in environmental biotechnology, because initially people were only seeing the functional elements, like whether a protein is expressed, people can study. But now with the study of the genetic makeup, it was found that the genes were flagella were present. On further doing some experiments, we were able to know that the flagella is produced under certain conditions and not in others. So we can exploit this theme in many of the microorganisms to customize the bioremediation process for a particular contamination site. So, and then apart from motility and flagella, then comes electron transfer and pili. So pili genes are specifically expressed when grown with insoluble Fe3 plus or manganese kind of electron acceptors. This can function as a nanowire and allows the extracellular electron transfer to iron molecules. This mechanism provides advantages over other iron reducing bacteria. So when we talk about anaerobic processes, electron transfer is one of the key mechanisms, and microorganisms in such environment are also syntropic, which is they depend on each other for their growth. So, and also there is involvement of extracellular electron transfer. Pili genes are not just the cellular appendages, they can act as the medium for this extracellular electron transfer. So, the interesting factor about its flagella and pili were discovered by genomic analysis. So with this, Geobacter has some competitive advantages compared to other iron-reducing organisms like Shivanella and Geothrix. Geobacter demonstrates superior efficiency, and this superior efficiency is because other bacteria produce energy expensive iron chelators to solubilize the Fe3 plus ions, whereas Geobacter uses direct physical approach through flagella motility and pili nanowires. This is one of the most energy efficient strategy for accessing the insoluble electron acceptors. So when there are two organisms competing, the fastest wins, and the fastness in Geobacter is due to the presence of flagella and pili, and all this we were able to discover with the advent of genomic analysis. So Geobacter is a versatile organism. It can be used in a single contaminant system, as we saw for uranium remediation or hydrocarbon degradation. It can also simultaneously remediate both uranium and hydrocarbon. It can enhance by bioremediation through acetate injection, because acetate is easily consumed by Geobacter. Henceforth, there is rapid electron production and Acetate act as a very good electron donor. It yields energy through the electron transport to the terminal electron acceptors. All this happens rapidly, therefore we can see enhanced bioremediation through acetate injection. So we can also see in situ bioremediation applications in various DOE sites. The comprehensive understanding of geobacter through genomic analysis has revolutionized the entire bioremediation field itself. So it has thrown or shed some light on how the study of molecular makeup of a microorganism can be used for optimizing customized processes for bioremediation of particularly contaminated sites. So, and now we go to the sulfate reducing bacteria in bioremediation. When talking about the sulfate reducing bacteria, it is another group of microorganisms that is very essential in terms of bioremediation. And we can find these bacteria with high sulfate concentration, which includes marine environments and groundwater systems. So the sulfate reducing bacteria utilizes sulfate as the terminal electron acceptors, and the process involves hydrocarbon contaminants are first degraded into carbon dioxide and water. Carbon and electrons are generated, and so first of all hydrocarbon is degraded to carbon dioxide and water. In this process carbon and electrons are generated. So this is utilized by the sulfate reducing bacteria. So here the electrons are transferred to the membrane bound carriers. Final electron transfer to the sulfate as the terminal electron acceptor, and sulfate is reduced to sulfite as the end product. So it utilizes the hydrocarbon to produce electrons, and that electron is then used for remediation of sulfate to sulfite. So some of the key bacterial species in the sulfate reducing bacteria group includes Desulfobacula. It is effective in sulfate reduction and as well as hydrocarbon degradation. And next one is the Desulfobacter. So the preferred environments for the sulfate reducing bacteria is the marine environments with naturally high sulfate concentration, groundwater systems where sulfate availability can be enhanced, anoxic subsurface environments where other electron acceptors may be limited, and environments with nitrate reduction having thermodynamical limitation. So when we saw about the sequence of terminal electron acceptor usage, we saw oxygen followed by nitrogen followed by manganese, iron, and then sulfate. So whenever the organism cannot use either of the above components, it uses sulfate. Coming to the redox considerations, under strict anaerobic conditions, nitrogen reduction is thermodynamically limited. So in that case, sulfate becomes a good option as an electron acceptor, and here the sulfate reducing bacteria are able to actively survive. The enhancement strategies include we can add sulfate to a particular environment like groundwater so that the sulfate reducing bacteria can thrive there. And we can also work on creating other favorable environments for SRB to stimulate the degradation rate or to speed up the rate of the reaction. So the effectiveness in pollutant degradation when using sulfate reducing bacteria has can be talked upon using the process similarities. The degradation process by the sulfate reducing bacteria follow similar principles to other electron acceptor systems. So here, first of all, there will be some organic pollutant which will be utilized by the bacteria to generate carbon and electron. So that is why that organic pollutant is known as a carbon source and electron donor. This electron, it transfers through this electron transport system and finally gives to the terminal electron acceptor. In this process, the microorganism gains energy and we attain bioremediation. Some organisms can also lead to complete mineralization to carbon dioxide and hydrogen. So, as told, this process generates energy as well as simultaneously allowing us to remove the contaminants. So, the advantage of using SRB includes that it is effective in environments where other electron acceptors are not available. It is capable of handling complex organic pollutants. It is a sustainable and naturally occurring process, and it is compatible with in situ bioremediation approach. The applications include in cases such as marine oil spill remediation, groundwater contamination treatment, and industrial remediation where sulfate supplementation is feasible, or industrial byproducts with high sulfate content can be remediated using SRBs, and also co-treatment scenarios with other bioremediation processes is also one of the applications in which this sulfate reducing bacteria is used. So next we will be talking about, first we saw about geobacter, next about the sulfate reducing bacteria, and now we'll be seeing about dehalococcoids. So before talking about the dehalococcoids, we'll talk about the polychlorinated biphenyls. So because the bioremediation of polychlorinated biphenyls or PCBs and other chlorinated solvents represents a unique category where the pollutants can serve as electron acceptors, requiring specialized bacterial systems for effective remediation. So, in all previous cases, we were talking about the presence of some terminal electron acceptor like oxygen in case of aerobic conditions, or in case of anoxic and anaerobic conditions we had nitrate, sulfate, iron, etc. But in the case of the presence of PCBs, they can act as the electron acceptors also. So the nature of this chlorinated pollutant is that they are significant contaminants at many DOE sites, and they are characterized by the chlorinated organic structures that are highly persistent in the environment. So, because of the presence of chlorine or chloride, or because of the chlorination, these structures are very stable and highly persistent in the environment. They can serve as the terminal electron acceptors in anaerobic processes, and they resist conventional aerobic degradation pathways. So, we need some reductive processes for effective remediation of this chlorinated contaminants. So dehalococcoids represents a special group of bacteria. It has been isolated from contaminated sites and bioremediation location. So the pure culture isolates have been extensively investigated for its capability to process the chlorinated compounds. So the mechanism by which dehalococcoids go for reductive chlorination is the utilization of fluorinated compounds like polychlorinated biphenyls, where these compounds act as a terminal electron acceptors, followed by sequential removal of chlorine atoms through reductive dechlorination. So first it utilizes the chlorine compounds and then goes for sequential removal of chlorine atoms through reduction process. So that is known as reductive dechlorination, and conversion of highly chlorinated compounds to less chlorinated or non-chlorinated compounds. So this is the final output that we are getting. It converts the chlorinated contaminant into a less chlorinated or non-chlorinated product. And finally, it obtains energy through this electron transfer process. So this is how dehalococcoids are used for remediation of sites with chlorinated contaminants. The application and the scope of dihalocolloids include that they can be used in polychlorinated biphenyl contaminated sites, chlorinated solvent treatment sites, and it can also be used in mixed contamination scenarios where chlorinated contaminants are also present, and it can be working in both aerobic and anaerobic sites. The process requirements for effective reductive dechlorination includes the provision of appropriate electron donor, maintenance of anaerobic condition, optimization of pH and nutrient conditions, and potential co-substrate addition to support bacterial activity. So these process requirements we have already discussed with all the microorganisms that we have studied. So now we will just have a little comparison about the metal reduction that we saw in geobacter and dechlorination that we saw in dihalocolloids. So here in metal reduction, the electron flow is from the organic donor to the bacteria and finally to the metal acceptor. So the electron flow from uranium 6 plus to uranium 4 plus follows electron donor to bacteria to metal acceptor. The result is that the metal is precipitated and organic compound is oxidized. So this is about the use of hydrocarbons and transformation of uranium 6 plus. So here the energy source is the oxidation of electron donor. In case of reductive dechlorination, which is mainly done by the dihalocolloids, the electron flow again follows the same pattern where the electrons from the electron donor goes to the bacteria and then to the final chlorinated terminal electron acceptor. So in this case, dechlorination happens and the toxicity or the harmfulness of the contaminant is reduced. And here the energy source is the reductive removal of halogen substituents. So that is the main difference in, or main distinguishing feature in dihalocolloids. So until now we have seen some basics about bioremediation and few examples like geobacter, dihalocolloids, sulfate reducing bacteria and all. We have seen the evolution of molecular techniques and microbial study techniques. So if you have any particular doubts until this topic, you can post it in the chat box. I'll just pause for a couple of minutes to see if there are any doubts, and then we'll proceed with the remaining uh, topics that we have to discuss in today's session. If you have any doubts, please post in the chat box. I'll just wait for a couple of minutes and then we'll proceed. Yes, uranium 4 plus. No, the concept of converting uranium 6 plus to uranium 4 plus is removal of uranium from the water system. Let's when we talk about some water system, if uranium 6 plus is present, it has a higher solubility in water and therefore it is persistent in that water. But when you convert it to uranium 4 plus and when the pH is near neutral, this uranium 4 plus tends to precipitate. Therefore, we can isolate uranium from the water system. So that is how we are using biotransformation to eliminate the contaminant from the contaminated site. Not only in uranium, in many metals, one of the oxidation state will be um, less mobile. So in one oxidation state, the metal will be highly soluble. So this solubility is very tough because when it is in a soluble state, we can't remove that metal from water systems, and it can also leach to nearby systems. When we talk about water systems, especially, the high solubility is highly dangerous because we can't remove that metal from that water system. Another uh, aspect of seeing the solubility is that when there is consumption of that water, again it should be highly soluble in inside our body also. So it can be transferred to our body and it can freely move in our body. So when it is highly soluble, it poses a higher risk of damaging the body parts, especially kidney, uh, when we talk about metal contaminated waters. So this concept is common to many metals. That is why we are trying to change the oxidation state of the metal, precipitate it and take it out of the system. Similarly, when that goes into the body, it will not have that much solubility or mobility inside the body, also, like in the water stream. So, that is why we can call it a little less toxic, but still it is toxic. Okay. So, with that, the next small topic that we will be seeing is about the 16S rRNA gene analysis in bioremediation. So, we now extensively know what is a 16S rRNA, why are we using it, and what is the reason behind using it, and what is the result of the study uh, of analyzing the 16S rRNA, and where we will be using the results. All this we know. We'll just go through it once more. 16S ribosomal RNA gene based analysis was one of the revolutionary things that happened in bioremediation research. It provided the molecular tools to identify and study microorganisms both in pure culture and complex environmental communities. So, this eliminated the need for culturing. So, when we talk about individual isolate identification, isolation of bacteria from contaminated sites, PCR amplification of the 16S rRNA genes. So, we were able to do PCR because 16S rRNA gene had conserved region. So, we were able to design primers and perform the PCR reaction and get many copies of the genes. And then we went for sequencing and phylogenetic analysis for taxonomic identification and integration with traditional characterization method happened. When we talk about total community analysis, direct DNA extraction from the contaminated sites or the contaminated environment was done, followed by PCR amplification, and then we go for clone library construction or direct sequencing. So the clone library construction, in case of Sanger sequencing, we first go for cloning the gene and then going for sequencing. Whereas in case of NGS, we can have a high throughput sequencing of many samples simultaneously at a time. And finally, we come to know about the composition of the community as well as its diversity. So based on this, we were able to trace metabolic reactions, metabolic relations between different organisms. All these we were able to perform. So 16S rRNA is a great revolution in bioremediation research, especially. And it can be used for both pure culture analysis as well as consortium analysis. Again, a repetitive slide where we talk about cultivation dependent, independent methods and traditional approaches. So the main reason why we needed a shift from cultivation dependent to independent method was because of the great plate count anomaly, which was identified by Staley and Konopka. So what that anomaly states is that only less than 1% of the total bacterial population is actually culturable. So when we go for studies based on culturing microorganisms, we are almost leaving out 99% of the organisms present, and we are studying only 1% of the bacteria that is present in the sample. So this means we are leaving out most of the organisms that are present, and we are not having a complete picture of the ecosystem. So what we came to know from this is vast majority of the environmental bacteria cannot be cultivated because we cannot replicate the exact natural condition in our laboratory, and organisms with competitive advantage or the influential organisms remain inaccessible through traditional methods. This creates a potential bias towards easily culturable rather than environmentally dominant organisms. So the easily culturable organism need not be the most uh, dominant or most widely present organism that is orchestrating the um, ecosystem. So the advantages of the cultivation dependent method is it gives us a detailed characterization where we have a comprehensive physiological and metabolic studies. We can do direct manipulation and genetic analysis. We can carry out experiments in controlled conditions and elucidate the molecular mechanism through which the microorganism uh, is functioning, and we can have pure culture of that organism. So we will have an extensive biodata of a single organism in case of cultivation dependent methods. In terms of application, the advantages is that production of bacteria inocula for field application, we can produce the pure culture. We can go for optimization of growth conditions and amendments. So this is about the research and development part. So when we have the organism in natural environment, when we are going to build a treatment system, we need to transfer the bacteria or the microorganism from that environment to our remediation site or the treatment system, which will be mostly a reactor. In that case, we need culture techniques so that uh, we have the active live bacteria that can be transferred from the natural site to our reactors. And we can, if we can grow these organisms in the laboratory, we can study optimizing the physical and chemical parameters to enhance the growth, to enhance the yield, energy gain, to enhance the rate of the process. So all this optimization is possible if the microorganisms can be grown in laboratory conditions. We can do different experiments, and we can uh, go for optimizing the process. We will also be able to develop standardized bioremediation protocols where we tell this much amount of inoculum of particular bacteria should be used for remediating a particular amount of pollutant. So all this standardization in terms of numbers can be done, and then we can ensure quality control and reproducibility in the applications of cultivation dependent methods. So here when we talk about the mechanistic understanding based advantages, we can directly observe the pollution degradation pathway. So this is based on chemical analysis, molecular analysis, biochemical analysis, all this we will be able to directly observe the pollution degradation pathway, and we can study which enzymes are responsible for this degradation and how the kinetics of this enzyme works. And based on that, as we talked about in the practical application section, we can go for optimizing the conditions to ensure the highest enzyme activity, and also we can take care that we do not employ any condition that will turn inhibitory for the enzyme or microbial activity. Both of this is possible. We can also go for genetic modification to enhance the capabilities. Like we saw, some genes can be non-functional, which we can functionalize, or we can insert some other advantageous genes from other organisms through metabolic engineering, and this genetic modification will again serve as a great addition to enhance the degradation process. And the last one is the detailed biochemical pathway elucidation. So pollutant degradation pathway and the biochemical pathway are two different things. Pollutant degradation in the sense, when we start with something like a dye or antibiotic, let's say we have uh, a methylene blue dye or some ampicillin kind of antibiotic. So, what are the steps in which this methylene blue or ampicillin is finally mineralized or transformed into a less toxic product that we can do? So, a compound A transforms to B and then to C, and finally it is mineralized as carbon dioxide and not, I mean, almost all the bioremediation process is a multi-step process. Directly from methylene blue or ampicillin, you don't get carbon dioxide and water. You have to go through a series of steps. So in that case, what are the transformations of the pollutant that is studied in pollution degradation pathways? Whereas in the biochemical pathway elucidation, the process, like we have biochemical pathways like glycolysis and Krebs cycle. So in that way, biochemically, what enzymes are used, how step by step the product is converted in, the pollutant is converted into a final product, what is the electron transfer or the redox reactions that is happening in the inside the particular organism, and how it is associated with the other metabolic pathways in the same organism. All these is about the study of biochemical pathways. So in that way we get detailed characterization of the organism. We have a mechanistic understanding of bioremediation, and we are able to develop the practical applications. So these are the advantages of cultivation dependent methods. But the major disadvantages is that many organisms are not cultivable, therefore we have limited representation, and we are at a high risk of isolating non-dominant and less influential organism and leaving out the dominant organism just because it is not cultivable. Coming to the cultivation challenges, we need complex nutritional requirements for organisms that are unknown. We will not be able to study the interspecies interactions or intermicrobial interactions, how one microorganism interacts with other microorganism, whether it is syntropic, whether it is competitive, whether it is parasitic. We will not be able to study any of the interdependencies within the microbial population. Uh, and in cultivation dependent methods, we will not be able to have a laboratory setup that exactly replicates a natural environment. And most important disadvantage in is the cost and time consumption. When we talk about cultivation independent methods, it allows comprehensive community assessment, and it follows a straightforward method. It has environmental relevance, and then it validates the capabilities of the microorganisms. So when we talk about uh, getting a comprehensive community assessment, we mean that the analysis of the entire microbial community without the cultivation bias is possible here, and we only need the direct environmental DNA and RNA samples for analysis. We can identify the organisms that were previously unknown, and then we can go for assessing how the community works. So, how different microorganisms act with each other in a particular environment, and when there is some shift in the nutritional or environmental condition, how would be the dynamics of the environment. All this we can have a comprehensive assessment of all of this. So when we talk about methodological straightforwardness, it is not about having some media trying to grow it for days and then performing some experiment. We just directly isolate the DNA, amplify it, and then sequence it. And based on the sequencing, we start profiling the community. So this is less time consuming and straightforward. So when talking about the environmental relevance in case of culture dependent methods, we are creating an artificial environment. So we don't know if naturally the organism is going to act in the same way. So direct analysis in their natural state will be more relevant for developing bioremediation processes. And then assessment of community responses to environmental changes, monitoring of bioremediation progress in real time, and identification of key players in natural attenuation. So all this is the environmental relevance based advantages. And finally, talking about the validation capabilities, we can confirm that the isolated organism represents the uh, real environmental condition, and we will be able to study the relative abundance of microorganisms. So, which is dominant, which is less present, even though there are more number of microorganisms present based on the activity which is dominant. All this we will be able to study, and we'll be able to track the community dynamics in result to the changes in the environmental conditions. When we perform both culture dependent and cultivation independent method, we can have a validation of the culture dependent method. So all this together fills the gap or fills the disadvantages of the cultivation dependent method. Coming to the disadvantages, it gives us limited functional information where the phylogenetic identification is possible without functional confirmation, and we cannot directly study metabolic capabilities. It is limited to the understanding of organism interaction, and it is very difficult to predict the bioremediation potential. In case of technical limitations, as we saw in the last lecture, we have PCR bias and amplification artifacts that can limit um, the results that we are getting. We can have DNA extraction efficiency and sequencing depth and coverage limitation and quantification challenges. So all these technical limitations were discussed extensively in last week's session. And coming to the application constraint, we cannot directly produce organisms for field application. We can only go for culture dependent methods to prepare an inoculum for reactors, and we have limited ability to optimize the growth conditions. Only in laboratory setup we can perform optimization experiments, and we will have difficulty in developing targeted bioremediation strategies, where we use only single organism, like we only go for a geobacter that can do this uranium reduction or biotransformation. And we also have challenge in translating the community data to practical application. So next comes the metagenomics approach. Now we have seen about cultivable and non-cultivable methods. Here comes the Metagenomics. So metagenomics is again like 16s rRNA. This is again a revolutionary approach to overcome the limitations of both cultivation dependent and uh, cultivation single gene molecular techniques, and it gives us comprehensive insights into the community structure, function, and interactions in bioremediation systems. So in the previous slides we saw there were different disadvantages, and metagenomics was one technique that could address disadvantages from both the systems. So here 16s RNA gene analysis has some limitations which we saw already, like phylogenetic limitation. It cannot reveal the interactions and dependencies. It fails to explain the community dynamics, and it does not give enough understanding about the complex processes. So all these disadvantages we saw from 16s rRNA gene analysis. Traditional approaches also have their own disadvantages, like not replicating the natural scenario. So that is why we go for metagenomics approach. Apart from genomics, we also have other advanced omics approaches. So in case of metagenomics, it is the analysis of total DNA. So whenever we talk about some meta-omics approach, it is going to be total DNA, kind of the word total is going to come along whenever there is meta. When we talk about genomics, it is about the study of DNA. So metagenomics, we talk about the analysis of total DNA. We have complete genetic inventory of the communities where we can identify the functional genes and pathways and assess the metabolic potential of the organism. So these three insights we can obtain from metagenomic analysis. When we talk about metatranscriptomic analysis, it is the analysis of total mRNA. So here, whenever there is some gene that is present in the metagenomics approach, when we can identify some gene, it is not necessary that that gene is expressed, but only when a gene is expressed, we can confirm the active functioning using that gene. Okay, so in that case, to study the functions, we go for the metatranscriptomic analysis. So only when a gene is going to be expressed into a protein, then only this mRNA is produced. So it tells us about active gene expression patterns, and not necessarily all genes in the DNA uh, need to be functional. So using metatranscriptomics, we can find which are the active genes that will be expressed, and we will be able to know the real-time community activity. Based on that, we will have the understanding of how the ecosystem works and how we have to design our bioremediation process. And finally, we can identify the responsive biological pathways. Like I told, we can't tell the pathway based on metagenomics, because metagenomics essentially does not allow us to tell which gene is expressed. So that is why we go for metatranscriptomics. And when we talk about metaproteomics, it is about the analysis of total protein pool. So we will know actually the express what are the expressed proteins and enzymes that are present, and what metabolic processes is happening, and we can give a functional validation of the genetic potential. So the applications of the omics approaches is it produces a comprehensive community function. So metagenomics ideally allows complete degradation, understanding of the complete degradation pathway, understanding about complementary metabolic capabilities. Like in case of syntropy, one organism should oxidize or perform reduction reaction in one step. The product of it is utilized by the next organism. So only when the first organism performs, the next organism will be able to survive in that um, environment. So this is one kind of a complementary metabolic capability which can be found out using omics approaches. We can estimate the community resilience and stability, how far it can withstand the environmental changes. So that is elucidated using omics approaches. And then we go for optimization of bioremediation process. Uh, Another thing is it can have a say about the predictive capabilities. So the prediction of bioremediation outcomes. Why? Because the omics approaches are again high throughput. We have more data. We can use modeling approaches to predict the bioremediation outcomes, and we can identify the rate limiting steps and try to uh, sort out the rate limiting factors. Like in case of anaerobic digestion, either the first step of hydrolysis or the last step of methanogenesis can be the rate limiting step. So we can see how to enhance the hydrolysis process or how to enhance the electron transfer process. So that way we can identify the rate limiting step and then try to find some remedies to overcome the rate limitation. And we can also go for assessment of treatment sustainability and design of enhanced bioremediation strategy. Coming to the monitoring and assessment applications, we can have a real-time monitoring of the bioremediation progress. We can assess the community health and stability, and we can also study the capability of the organism to degrade emerging contaminants, and we can study the treatment effectiveness. So that is the our final motto, to study how effective we can carry out the treatment process. So cultivation dependent, independent, and omics approaches. I hope it is clear. So next we will talk about the multi-layered complexity in bioremediation. So the concept of multi-layered complexity represents a shift from traditional reductionist approach to systems level understanding of bioremediation. So reductionist approach in the sense isolating one organism, studying it, and then generalizing its function to the entire ecosystem is the traditional reductionist approach. From that, we have to study, have a systems level understanding of bioremediation. We have to understand what are the multiple layers that are present, how the presence of these layers is decided, and how the interactions between these layers determines the overall outcome uh, of biodegradation or bioremediation in a particular habitat is very crucial, and that is what is represented as a multi-layered complexity concept in bioremediation. So this recognizes that the environmental remediation involves complex interactions across multiple organizational levels. So the limitation of reductionist approaches, it has traditional assumptions like many bioremediation studies have been dominated by simplified models because we have assumed that one bacterial strain is the key player, one compound is a primary target, and one pathway can fix the issue. We are building a linear cause and effect model. So we study the ecosystem, we fix that, okay, pollutant A is the primary target. If you are eliminating A, then the problem is solved, the pollution or the contamination is removed, or remediated. And we go about thinking that, okay, this bacteria, if a bacteria X can act on A and mineralize it, okay, then bacteria A is the key player, we don't need anyone else. So a direct interaction of A and X solves the remediation problem. And ideally, when we have one organism and one pollutant, it is a single pathway. But this is not how things work in nature. We try to form a linear cause and effect relationship, but it is a very complicated networked relationship that works in natural environment. The reductionist approach often fails because uh, environmental systems involve multiple interacting components, and complex feedback mechanism exists between these levels. From, as we talked about this multi step degradation process, each step transforms the primary contaminant into some other component. but the component that is coming as a product can be more toxic than the actual contaminant we were trying to remediate itself. So that is also a key factor that we should consider, and that is why we should go upon chemical analysis to elucidate the pollutant degradation mechanism. So this reductionist approach, which simplifies the entire complexity of the bioremediation process, needs to be removed. So that is where the concept of multi-layered complexity or multi-layered organization structure comes into picture. So bioremediation systems function across multiple interconnected levels. First one is the molecular level. Molecular level in the sense genes, enzymes, operons, and pathways. So genes encode enzymes responsible for pollutant transformation. Ultimately, the component that is going to directly attack or directly interact with the pollutant is going to be enzyme. Then why do we need the microorganism? Because microorganism is the one that is producing the enzyme. And how does it produce? Because of genes. So there is this DNA and it has multiple genes. Each of the gene can code for a particular protein, and if that gene is functional, the functionality of the gene can be affected by the nutritional requirements as well as the environmental conditions. So all this understanding we need. That is why we are studying the gene, to see whether it has the potential to produce a particular enzyme that can degrade the pollutant. And then we study about the mRNA to confirm the functionality, and then we go about studying about the enzymes to see how we can further enhance the kinetics of the enzymatic process. And we all, from all this analysis, we try to construct the pathway. We try to see if there are some operons present in that. So if then what are the conditions we need to uh, supplement for the pathway to take place or the operons to be functional. So this is about the molecular level organization. Next comes the cellular and population level. In this, we study the genomes, which is the complete genetic blueprint of the organism, and then go about studying about the cells. So inside the cells, we have the genome. First, we studied about the genome. Further, when we zoom out, we come to the cells, which are the individual functional units containing all the genetic and enzymatic machinery. So the cell is the one that encompasses the genome as well as the enzymes. Uh, Further, when you zoom out, you have the species, where similar cells together you call as species, the group of genetically similar organisms with shared capabilities, as per the definition here. And finally, when you again zoom out, you have populations where you have different species that are present. So inside a cell, there is a gene, you have one cell, you have a group of same cells. Similarly, you have a group of different same cells. So that is how, if you imagine zooming out a picture in your mobile phone, you can see start from the genome, zoom out to cells, to species, and then to population. So this is a cellular and populational level organization. And next comes the community and ecosystem level organization. So when we talk about communities, multiple interacting species with complementary functions can be identified. So in the previous subtopic, we talked about until population. Now we talk about community. So we were studying about the interaction between these species. And then comes the idea of niche, where the specific ecological role as well as its resource utilization pattern, how it uses organic compound or inorganic compound present in the environment. All this is comes into picture. And then comes habitats and microhabitats, where we talk about the physical and chemical environments that drive the microbial metabolism. And finally, we talk about sites, which is the location which is contaminated and requires bioremediation. So starting from genes to the sites, you just have to zoom out. to visualize each of this level, and this is known as multi-layered organizational structure. So when we talk about multi-layer organizational structure, there is complex interaction network between these layers, and that is the key phenomenon that is making us study the multiple layers. So what kind of interactions can we expect? We can expect vertical interactions and horizontal interactions. So in case of vertical interactions, it gives information and influences both up and down the organizational hierarchy. So we can study genetic expression influences enzyme production. Enzyme activity again affects the cellular metabolism. Cellular activity impacts population dynamics. Population changes influence the community structure, and community function affects the ecosystem. If you just see the previous slide, whatever components we have talked about, each of it is contributing to the next. So if one is affected, then entirely until this sites everything is going to be affected. So at least here we have until the communities, right? So when gene expression is influenced, it affects the enzyme production and activity, which in turn affects the cellular activity itself, which in turn uh, leads to population change and a disruption in the community function. So this is a vertical interaction. When you talk about horizontal interaction, the interaction is within each organizational level. So vertically, when we talk, it goes from one to the next, from genes to cells, genes to enzymes to cells to populations to community. It is vertical. When we talk about horizontal, it is within that organizational level itself, where we have gene to gene regulatory networks, enzyme to enzyme, cell to cell communication, species to species competition, and community level resource sharing and niche partitioning. So, this is the horizontal interactions. So, the presence of one gene can silence another gene. This is one example to remember gene to gene regulatory networks. Similarly, when you read about the concept of operons, you can know how gene to gene regulatory networks work. Enzyme to enzyme metabolic coordination is required, and that in turn is affected by the environmental conditions also. And finally, there is cell to cell communication, like we have phenomenon like quorum sensing and all, where cell to cell communication affects the functioning of the ecosystem. We have species to species interaction, which is mutualism and competition. Like I said, in case of syntropy, the organism A needs to produce some product for organism B to utilize and then produce another product out of it. So that is syntropy. In case of competition, one microorganism is trying to outpower another. So each has to be evolutionarily more active to survive or thrive in that environment. So such species to species interaction happens. In community level, we have resource sharing and niche partitioning that we can observe. So this is the multi-layered organizational structure, and this is the different interactions that we can find in the multi-layers. So when we talk about this interaction, what are the environmental control factors that determines this interaction? So the first is the abiotic landscape. So when we talk about abiotic landscape, we are not talking about any biochemical activity or microorganism here. We are just focusing on factors like physical factors like temperature, humidity, and pressure, chemical factors like pH, redox potential, and conductivity, nutritional factors like the presence of carbon, nitrogen, and energy source, or the presence of substances like phosphorus, or the presence of other terminal electron acceptors. All this comes under the nutritional factors. And then the matrix conditions, which again is a kind of a physical factor like the soil texture, porosity, and water content. And next comes the pollutant characteristics. The pollutant characteristics include how chemically diverse the pollutants are and what is the mixture effects. Like when we have a site that is contaminated with both some um pollutants like antibiotics as well as some complex other pesticides, then the situation is going to be worse. We cannot focus on only degrading the antibiotics or the pesticides. The interaction effects can be even more dangerous, and it can be slightly different. We can't straight away apply the single component system mechanism to a mixture effects. So that is one point. And then the concentration gradient and spatial distribution within the contaminated site. In some places the contamination can be of a higher magnitude, whereas in others it can be low. So such pollutant characteristics we should be aware of. Bioavailability variation, like I told, when we have some metals and all, some metals can be easily bioavailable, some cannot be bioavailable. So this trade off for uh, biological means of remediation is very important, and we should have a knowledge about transformation product formation to ensure that we are not degrading the pollutant into a more toxic component. So we need to have a bidirectional control here. Environmental conditions control the microbial composition and function. Biological activities modify the environmental conditions, and this feedback loop creates the dynamic equilibrium. Environment affects the microbes, microbes affect the environment. So this is a feedback loop. And when both this is functioning properly, then we have a dynamic equilibrium. So what we refer to as a dynamic equilibrium is there is continuous degradation happening. The microbes are very active and the continuous degradation is happening, but there is the contaminant is getting reduced. It is getting produced and it is getting reduced. So that gives us a dynamic equilibrium. And then system responses emerge from these complex interactions, so which we have as a result of our systems level analysis. I hope it is clear up to this. I'll just wait for 2 minutes to see if there are any doubts, or we'll proceed. Okay, then we'll go to the next slide where we talk about the bioremediation space concept. So we have different landscapes when we talk about bioremediation. The first one is the catabolic landscape. Here we have degradation pathways available in the community. We have enzymatic diversity and specificity. We have metabolic flexibility and adaptation and biochemical transformation potential. So essentially, catabolism is happening here. For that, we need enzymes. Our environment is not a single pollutant system, so we need enzyme diversity. But we need enzymes that can target each of the pollutant. So we need diversity and specificity hand in hand. It should specifically target the pollutant, and for each of the pollutant, we need a enzyme. So that is where the concept of enzymatic diversity and specificity comes into picture. This happens only when there is the degradation pathway that is available in the community, and it should not be very rigid because the environment keeps on changing. So it has to have a little bit of metabolic flexibility and adaptation. This finally sums up as the biochemical transformation potential of that community. So this is the catabolic landscape. When we talk about the chemical landscape, we are referring to the pollutant characteristics and distribution, what pollutant is present and how it is distributed. It can be concentrated at one point, diluted at the other. So all this information we need. We need to know the nutrient availability and limitation. So the pollutant can itself act as the carbon energy source. If not, we need some other carbon energy source for the organism to first survive and then go about remediating the pollutant. We need to know about the geochemical processes and interactions. So how the presence of the components in the habitat itself, the abiotic factors as we saw, affects the remediation process, and we need to estimate the mass transfer and bioavailability factors. So this is about the chemical landscape. When we talk about dynamic interactions, it is both. Catabolic capabilities determine the pollutant phase. So catabolic landscape uh, directly influences the chemical landscape, and the chemical conditions are the reason why certain microorganism is thriving. Let's say we have a acid mine drainage. So in that case, the pH is going to be very low. So acidophilic organisms will only be able to survive. Acidophilic organisms are able to survive because they have mechanisms to prevent against such low pH by still utilizing the energy and carbon resources in that particular environment. So this trade-off is what is mentioned as a dynamic interaction, where catabolic capability determines the pollutant fate and the chemical conditions determines the presence of microorganisms in an environment. Biological activities modify the chemical environment, and the entire study is going to be meaningful, and bioremediation is going to be successful through the co-evolution of biological and chemical systems. So when we talk about the multi-layer assembly, it can be stated in the context that we saw previously or using this catabolic and chemical landscape terms also. So next we will see about the polyphasic taxonomy in bioremediation research. So polyphasic taxonomy represents a comprehensive approach for bacterial identification, and integrates molecular and biochemical techniques. Apart from 16S rRNA analysis, so this gives us more accurate and it gives us highly resolved information for bioremediation microbiology. It combines independent analytical methods to create a comprehensive taxonomic framework. So we are going to perform multiple analysis, have the information or data backup where one data supports the other, and through this study we will have a comprehensive taxonomic framework that overcomes the limitation of individual techniques. Like when we have some material or compound, we go for different analysis like imaging techniques like some electron microscopy. We go for things like XRD, we go for FTIR, which where we talk about the functional groups. All this we do just to confirm that it is some material. Similarly, we perform multiple analysis where the data of one backs up the other data, and we can come to a comprehensive conclusion from all the data that is available. And this is beyond single gene analysis, and this is again repetitive from uh, all the previous slides that we have discussed. So the components of polyphasic taxonomy include molecular markers and biochemical markers. So in case of molecular markers, the first one is the 16S rRNA genes. It is the primary phylogenetic marker for bacterial taxonomy or most eukaryotes, and we have universal presence so that we can just design a primer that is commonly used for multiple organisms, But in order to differentiate between the organisms, we study the variable regions. So it has the advantage of both conserved and variable regions. One is for primer design, the other is for species differentiation. And the next molecular marker is the single copy marker gene. Single copy marker gene is an alternative housekeeping genes with good phylogenetic resolutions. Housekeeping genes in the sense genes that should be commonly available in most of the microorganisms to carry out the day-to-day activity of the microbial cell. They are genes with essential cellular functions and low horizontal gene transfer rate. Horizontal gene transfer is within the same generation. Vertical gene transfer is from one generation to other generation. Like we saw the vertical and horizontal interaction, vertical is going to be from one thing to the next thing. In terms of generational difference. Whereas horizontal is within the generation itself, from one organism to another organism is a horizontal gene transfer, and single copy marker genes are with low horizontal gene transfer rates, and they have complementary phylogenetic information and enhanced resolution for closely related species. When we talk about biochemical markers, we have this FAME analysis which we saw in the first slide. It is fatty acid methyl ester analysis. It analyzes the cellular fatty acid composition and gives data about species specific fatty acid profile. And then chemotaxonomic identification, it is a chemotaxonomic identification method, and standardized databases are available for comparison, and it provides rapid identification capability. We also have lipid markers where we analyze the membrane lipid composition, which is the phospholipid fatty acid profiling, PLFA profiling, which can act as species and growth condition indicator. It can also tell us about the environmental stress. So it is called as environmental stress response markers, because based on environmental stress, the lipid composition is going to vary. So that is why it can act as marker, and it is an indicator of the community structure. So in case of polyphasic taxonomy, we use multiple techniques, and we mainly use molecular markers and biochemical markers for identification. So next is a small topic where we see about what is biodegradation. Biodegradation is a biologically catalyzed reduction in the complexity of organic compounds. So we are just going to reduce the complexity of organic compounds using microorganisms. It involves the breakdown of the complex structures, which results in release of electrons. In any site, there will be some indigenous microorganisms that will naturally live in that particular environment because it has the capability to survive uh, or metabolize in that environment. So biodegradation process can either result in the complete breakdown, or it can lead to a biotransformation kind of a process. So the metabolic requirements for biodegradation uh, is energy source, carbon source, electron donor, and electron acceptors. These priority requirements forms the basis of metabolic diversity encountered in nature. When microbes are going to interact with the organic pollutants, they produce degradative enzymes and they carry out redox reactions. And these redox reactions are not single step reactions, they are going to be a series of biochemical reactions. And this degradation process is known as catabolism. And during this process, electrons are extracted from the degradation of the pollutant or from the organic compound, and it is transferred to the electron carriers, which further undergoes electron transfer. So now talking about the electron transfer or electron transport mechanism, it is operating through a membrane bound electron transport system. which we call as a electron transport chain also, ETS or ETC, and it is very crucial in bioremediation. The electron carriers transfer electrons to the terminal electron acceptors. We saw the order like oxygen, nitrate, iron, sulfate, and finally carbon dioxide in increasingly anoxic conditions. While electrons are transported through this electron transport system, they create a proton motive force. So, because of this proton motive force, I think in week 2 or 3 session we saw about this electron transport system and the proton motive force and all. There is going to be some proton gradient, and we have ATP synthase which can produce ATP and other forms of energy for cellular processes. So this essentially occurs in two pathways. One is the oxidative pathway where microorganism oxidizes the organic pollutants to carbon dioxides or other intermediates. Or it can also be a reductive process. So here uh, we have pollutants like chlorinated solvents or nitroaromatic compounds which serves as electron acceptors itself, and hence they are detoxified. In this case, microorganisms will need organic matters to act as carbon and energy source while reducing the chlorinated or nitroaromatic compounds. In case of oxidative processes, the microorganisms can utilize the pollutant itself as the energy and carbon source. In reductive processes, apart from the pollutant, it needs some organic matter to act as an electron donor. So next comes the concept of catabolism and anabolism. Metabolism is a combination of both catabolism and anabolism, where catabolism involves breakdown and oxidation of organic pollutants, and anabolism encompasses biosynthesis reactions. So catabolism is breakdown, anabolism is building up. Both are opposite reactions. During the oxidation processes, electrons are extracted and transferred to electron carriers. This simultaneously happens with breakdown of larger molecules into smaller intermediates. So that is catabolic processes. It produces substances such as pyruvic acid, phosphoenol pyruvate. All these are intermediates in glycolysis pathway. We all can also produce acetate, which can serve as precursors for macromolecules biosynthesis. In case of anabolism, they are energy requiring reactions. So catabolism, ideally when we are breaking down, energy will be released. So they are energy synthesizing reactions. In case of anabolism, it is energy intensive. The energy that was produced during catabolism can be used during anabolism to build up cellular components. So the interconnection between these processes is very essential for cellular growth and proliferation. Microorganisms deploy degradative enzymes for catabolic reactions while simultaneously facilitate anabolic reactions through biosynthesis enzymes. There is clear distinction between the role of enzymes in the microbial metabolic pathway itself. So next we'll talk about the concept of mineralization. So mineralization represents complete biodegradation, where we are not talking about any intermediate. We are seeing that the pollutant is completely degraded into carbon dioxide and water. Like when organic compounds contain sulfur or phosphate, they are also released and subsequently utilized by other cellular activities as nutrient sources in their mineral forms. Like DNA synthesis needs phosphates, or ATP again is adenosine triphosphate, and sulfur is required for some other cellular activities. So they are completely utilized in that case, they are not left. the process where nothing is left over, everything goes back as carbon dioxide, water, or other consumable molecules, is mineralization. The mechanism of mineralization includes it occurs with central catabolic pathways, the intracellular biochemical reactions of the organisms, and then transforms the organic compounds. This frequently occurs with aromatic hydrocarbons and aliphatic hydrocarbons through central carbon metabolism, and the biodegradation intermediates become precursors or components of the central carbon metabolism. This allows them to enter the central metabolic pathway and results in the complete breakdown of that organic molecule. And finally, we will see about the key differences between biotransformation, degradation, detoxification, and activation. Either of this mechanism can be used in bioremediation. Biotransformation is one of a umbrella term or a broad term. It means that there is some alteration of molecular structure or oxidation state of a chemical, and when that is transformation. And when we talk about biotransformation, this alteration in the molecular structure or oxidation state is brought about by microbiological or enzymatic methods. It includes two aspects, which is alteration of molecular structure and change in oxidation state. In an example of alteration of molecular structures, it converts long alkane chains to small acetate molecules, or it breaks benzene rings to catechol. All these are examples of alteration of molecular structure. Change in oxidation states we have seen in case of geobacter, like uranium 6 plus to 4 plus, chromium, hexavalent chromium to trivalent chromium. Similarly, in case of arsenic or many metals for that matter, we can see change in oxidation state. These alterations generally decrease the complexity of intramolecular bonds, and in very rare cases, they can also increase the complexity. So, when we talk about biodegradation, it is a more specific term referring to biologically catalyzed reduction in molecular structure complexity of organic pollutants, where they break down the molecular bonds, resulting in simpler structures. It's just going to degrade it. In transformation, in some cases, we can see that the complexity is increased, whereas in biodegradation, we only see the resulting of simpler molecules from complex molecules. This process can involve subtle changes or severe changes. So, in case of subtle changes, we can just observe removal or substitution of functional groups. In case of severe changes, we can see complete breakdown into carbon dioxide and water. So, the process of detoxification occurs when the biodegradation of the priority pollutants results in products of lesser toxicity. We are not getting any simple molecule, but just the toxicity is reduced. So, in this case, biodegradation is synonymous to detoxification. However, biodegradation does not necessarily lead to detoxification. So, this is a key point that you have to remember when you read about biodegradation and detoxification. Next one is the concept of activation. So, activation represents the opposite of detoxification. Here, the biotransformed product exhibits greater toxicity than original compound. In this case, biodegradation leads to the activation of the molecule, and it creates more hazardous intermediates. These activated molecules undergo another round of microbial attack, leading to further degradation. So first it like increases the toxicity, and then it further acts to decrease or degrade the organism. This process is crucial to consider when assessing risks associated with bioremediation implementation, particularly in mixed waste environments or inadequately characterized contaminated sites where some compounds might be activated, leading to increased toxicity or remediation hazards. So, with this, we come to the end of this session. on bioremediation and biodegradation. If you have any doubts, please post in the chat box. Otherwise, we can close the session. Thank you for joining for today's session. I guess we can close the session. Thank you.